I'm calling this Getting to IND for the rest of us because try as we might, not all of us are going to land in Cambridge or Palo Alto. And this is a little bit of my, a little bit of an experience talk on my tortuous path. But first of all, some ground rules. I have an executive position as the Executive Vice President and Chief Scientific Officer of Incisus Therapeutics, and some of the information I'm going to talk about are related to trials that we have and studies that we have in progress. I'm going to be discussing our first IND, which is a fairly simple one, a clinical trial that's designed to supplement immune function with in vitro expanded and activated gamma delta T cells after haploidentical transplant and post-HSCT cyclophosphamide. And this is a lessons learned talk, not a how-to talk. The FDA has published detailed guidance and regulatory and validation processes that are required for phase 1 cell and gene therapy IND. And I'll emphasize this as we get further along in the talk. And also just references to my former employers, such as Palmetto Health, University of South Carolina, University of Alabama, Birmingham, are specific to the time period discussed and may not reflect the views and policies of the administration at this time. This is the obligatory forward-looking statement that I have to show as CSO of the company. And we're providing this information in this release of this date. So our intended audience, if your laboratory looks like this, you probably are not going to benefit a lot from this talk, but you're welcome to stay and listen or go to the posters or whatever. If your laboratory looks like this, I think you're going to learn a lot from what we have to discuss today. So what are the building blocks to a successful IND application? Let me back up. I think I had one. Yeah, no, okay. In vitro data, basic animal efficacy data, if you need it, animal safety and efficacy data, GLP, good laboratory practice or closer, and an immunocompetent model, and if possible, if you have the tumor, a human patient-derived xenograft is also very helpful. You should show the FDA a logical pathway to GMP manufacturing of effector cells and vectors that details efficacy, purity, potency, sterility, and for cells identity, and exhaustive and detailed documentation of primary and derived data. This is important for you postdocs as well, that you got to write it, and graduate students, write it up every day, and as time and date stamp it, so you'll really enjoy coming back to that in the future. And of course you need the infrastructure to perform the required manufacturing and clinical therapy. So here's a little bit off the subject, but pre-IND for the undergraduates and graduate students and postdocs here. Bet your next mentor. Your career depends on it. If you're going to get into the translational research game, you need to find out where are his or her students now? How many national meetings have they attended as a speaker recently? Cross-institutional collaboration, what's their R01, PO1 score, consistency and quality and impact of publications. And I'll stop here and say that a clinical translational researcher like myself, frequent publications in molecular therapy, blood, neuro-oncology, those types of things are considered very good. If you go back to basic translational, you want to see a smattering of Nature, PNAS, those types of things. So, you know, look at what you're judging. And also, are you part of a T32 or other mentored student program? Is your prospective mentor or junior faculty? That can really be exciting, but you have to take this a step higher and see who's talking to them. Second thing, get your work out there. Small and high impact meetings, AACR specialty meetings, Keystone Symposia, and national meetings in the field. Sometimes you even have to pay for it yourself. A couple of personal things here. One, I didn't know any of this. I have literally, depending on your religious persuasion, either by the Lord's will or by dumb luck, I fell into Adrian G's lab that had a whole, about 30 junior faculty, postdocs, and graduate students and technicians that were way smarter than me. And basically that opportunity launched my career. And again, my first time I ever presented this gamma delta T cell data was at a Keystone meeting as a talk and abstract in 1996. And I think that we got to know a lot of people there who had never heard about this work before. So that was helpful as well. So it's an experience talk. Like I said, and now some science. Okay, these are slides from my presentation at ASH in 1998 to show you a little bit where we started. We were transplanting patients with haploidentical grafts back in 1992, 1993. We had a protocol that required total body irradiation, rather strenuous total body irradiation, and a very tough conditioning regimen, VP16, cytosine arabinocyte, cyclophosphamide, and methylpred. We took the T cells out, the alpha beta T cells out with an antibody called T10B9. It was an IgM that we manufactured in mouse ascites, and then we lysed the cells with complement. The FDA would not let you do that now, but that's what the technology was at that point. Had a very oppressive immunosuppression regimen as well. Took cyclosporin out for a year and ATG out for 10 days, and a long methylpred steroid program. So what we began to see was a bump in gamma delta T cells up around day 180. It topped the 10% mark, which was what we thought would be the highest number, the highest percentage of cells that we would ever see. And when we looked at that more closely, we saw that it wasn't everybody that was doing that. It was about a quarter of the patients who were developing very high gamma delta T cells, very high numbers of gamma delta T cells, and sustaining them over time. That little black line down there at the bottom is what we would consider the upper limit of normal. And the other interesting correlation was they weren't dying, the patients that were developing the high numbers of cells. And we didn't see a death till 13 or 14 months. And these patients are different. These patients, back in 1993, if you had leukemia, if you got AML now, you get induced, consolidated, and transplanted. You don't get a chance to relapse. These days, those days, these children were given 3 or 4 lines of chemotherapy before they finally decided to transplant them. And if they had a haplotransplant, they were really in a special category. So the fact that these kids were living as long as they were was fascinating. And also children that recovered with normal alpha beta gamma delta ratio were showing a death pattern that was similar to what we would expect with these types of transplants in this type of disease. We showed our biologic correlates. The first graph, the blue line, is our gamma delta T cells separated from one of these patients who grew high numbers of them against K562 cells without any further stimulation or culture. The darker line at the bottom are actually my gamma delta T cells that I sorted and figured I wasn't sick at the time to see what they do. So these cells were circulating and were activated. They killed K562 cells, they killed Raji cells, and MOLT4 cells, which are all leukemia lines, but they left alone normal donor mononuclear cells, third-party mononuclear cells, and EBV transformed blasts. In the last pattern, we see the gray are a mixed lymphocyte culture of purified gamma delta T cell, and the black is a standard mixed lymphocyte culture one way, and you see that the gamma delta T cells are not proliferating or taking up thymidine in the mixed lymphocyte culture. So we thought we had a cell that would kill leukemia and not cause graft versus host disease. And that was pretty exciting back then. So, summary and conclusions, increased circulating gamma delta T cells are early post-BMT is associated with significantly improved relapse-free survival with no additional GVHD risk. The infusion of alpha beta T cell depleted graft into a ablated host is associated in some cases with homeostatic reconstitution of gamma delta T cells. The predominance of the cells is consistent with an upregulated cytokine environment. Post-BMT immune system, as we know, is substantially upregulated even despite immunosuppressive therapy. And the expanded gamma delta T cells from patients with their cytotoxic to leukemia and also to their leukemia cell lines, or their own leukemia cells, when we were able to have them frozen and bring them back up. So this was all done under the watchful eye of my mentor, Dr. Adrian G., who, when I asked about he was getting ready to leave and I was getting ready to move on to my first assistant professorship, asked him about the property of this project, and he said, "I have my career. This is yours." So great mentoring. By the way, he won the ISCT Lifetime Achievement Award in 1990, or in 2016, which was very deserved. So, just finishing up with this science, gamma delta T cells demonstrate broad recognition of tumor-associated stress antigens. This is different from alpha beta T cells, which see processed antigen that's presented by the antigen-presenting cell group. They differ from natural killer cells as they can immediately recognize and lyse malignant cell targets independent of class 1 status. They recognize stress-associated proteins by both the gamma delta T cell receptor and the NKG2D receptor. And gamma delta T cells do not initiate graft versus host disease, and they facilitate donor engraftment. Lesson 2, file your intellectual property early. Now this would seem apart from IND, but it's really not, because if you're going to run a clinical trial and you're going to ultimately have to take this into a company funding, one of the first things the company is going to look for is intellectual property. Build a case, because a lot of times your university administrators aren't going to want to shell out the money to prosecute the patent unless you immediately have a licensor. 3 criteria, I think most of the people in the room know, but it must be new, it must be useful, it must be non-obvious to one skilled in the field. And that's a hard one to do. Patents give the owner exclusive rights to a product for about 20 years, and they encourage companies to take greater risks and invest more funds into your research and development. IP protection will be essential for developing your work beyond the point of preclinical, and probably will be essential for funding your phase 1 trial. And the FDA will expect you to have a path from phase 1 to phase 3, even when you come to them with a phase 1. You're still looking at the regulatory imperatives of phase 1 trial, but the FDA does not want to review 20 phase 1 trials that never move beyond that. They want to see that you've actually thought this through and how it's going to ultimately get out to the wider world of patients. Lesson number 3, be prepared for things that you don't see coming, such as your infrastructure disappearing beneath your feet. I got called into a room one day with 90 other people in the midst of my research and fired, along with all of them, because the hospital decided it didn't want to do bone marrow transplant anymore. At the time, I was fortunate. I had about $1.6 million in NIH and Leukemia Society grants and other things, so I was able to find a job, but still it interrupted things quite a bit. So it's likely that your experience will not be as sudden and dramatic as mine. But changes occur over an institution's lifespan, and essential faculty come and go, course support services are terminated, institutional research focus changes, and so on. So if your dream is to bring your work to patient trials, you have to take every recruitment seriously in the light of your project development strategy. Be prepared to move on if you encounter negative changes, loss of institutional interest, unusually burdensome administrative barriers to advancing the work that is already funded. Now, I'm not saying that your administrators yells at you when you run out of money. That's normal and expected. But if you're sitting on, a nice grant portfolio and you still can't get anything done, that's a red flag. So we took the work to the Medical University of South Carolina down in Charleston, their pediatric bone marrow transplant program, for about 4 years, sort of as an interim parenthesis while we were trying to figure out what we were going to do. And there we completed work on T-cell receptor CD3 region sequencing, showed that the V delta 2 compartment of the gamma delta T cells was depleted in first diagnosis leukemia patients, and V delta 1 was preserved. V delta 1 also had several public sequences that we thought were quite interesting and still actually need to be followed up by somebody. We collaborated with progenitor cell therapy, PCT, on scale-up in engineering with a now extinct bioreactor called the Isolex, if some of you remember back then. The work ultimately failed because the demands of the final graft exceeded the capacity of the bioreactor. And we also identified and procured what ultimately became the anti-alpha beta antibody for T-cell depletion that allowed the project to move forward. If you ever want to corner me for a six-pack of beer and a couple of hours, this is a great story. But one I can't go into now, but it was getting that antibody was really something. We then moved to UAB was recruited there by Richard Lopez and Bill Vaughn, where we designed, built, and staffed a GMP facility and an immunodiagnostics laboratory. We identified our physician collaborators. We obtained FACT accreditation for advanced manufacturing. We re-educated our CCTS and Drug Discovery Alliance about the unique aspects of investigator-initiated cell and gene therapy and IND filing procedures. If any of y'all remember UAB back in 2004, 2005, they had a SLAM gene therapy program there led by David Curiel. There are some changes administratively that went on at the top, which resulted in a massive exodus of those people and several others, and sort of left us without any of that, really. And I was brought in to kind of rebuild some of those things, and we almost got there, actually kind of got there, so it's still a struggle. And we worked but when we filed this IND, we worked with a very enthusiastic and immensely helpful team. We had to educate them. They were used to CEDAR filings and not CBER filings, so we had to sort of educate them on that, but they were extremely helpful in the end. And we filed UAB's first cell therapy IND. So while this is going on, a young lady named Kamar Goder in my laboratory on her way to taking over pediatric bone marrow transplant at the Virginia Commonwealth, did a 8-year look back at these patients who had developed increased gamma delta cells and saw that the finding held. And the only statistic out there that was anywhere close to explaining this was a direct correlation between the number and activity of gamma delta T cells and long-term survival. So you have a finding. How do you turn this finding into a therapy? That's the crux of when you want to take your basic translational work and move it into IND. We needed to recreate the conditions of homeostatic reconstitution, which are lymphodepletion, infusion of the desired T cell effector population, and make it purposeful, not random as it was before. And there were barriers to this at the time. We didn't know whether to ex vivo or in vivo expand gamma delta T cells. We didn't have any practical method for expanding gamma delta T cells. We were doing it on feeders in the laboratory, at leukemia feeders, and the timing and the conditions of the therapy in the context of haploidentical transplant, nobody really thought about or understood or was willing to argue with you about then. So we had really a long climb to do to try to make this work. But the other thing that is great about science is that other people are working on things that are related to what you do too. So the lesson number 4 is to incorporate the work of others. There were 3 paradigm-changing discoveries that opened the potential for gamma delta T cell therapies. One of them was by Steve Rosenberg and Mark Dudley, who although we described the process of homeostatic reconstitution as a postulate in the discussion of our first paper, they proved it basically in the laboratory and executed studies where they were lymphodepleting patients and infusing TILs, seeing expansion of the TILs, and in some cases long-term remissions from melanoma. Johns Hopkins developed the cyclophosphamide-based in vivo T cell depletion protocol, where after they give the haplograft, on day 3 and 4, they give 60 milligrams per kilogram cyclophosphamide, which is designed to kill T cells in cycle, which actually are probably the cells that are alloreactive. So they took out the GVHD problem for the first 100 days after a haplotransplant and made this procedure cool again. They still run into problems with relapse, and that is where we come in later down the road. So, and also Yoshihiro Tanaka in Kyoto managed to figure out how to manufacture gamma delta T cells, so small molecules called isotomal pyrophosphates, and that led the way to being able to make high numbers of these cells that would be significant for clinical use. So we've had the goal in view. Not still far away, but we can actually see it. That's the tip of Everest, actually peeking behind the Neptune wall. I took that picture at the Tenzing Memorial on my way up to base camp. So now you're in business. You've got to figure out how to take this from a bench to production. And these are data we presented at the European Bone Marrow Transplant meeting in 2014, where we're taking this from a T75 flask to the Miltenyi Prodigy. Previous to the Miltenyi Prodigy, no other bioreactor was available or workable for gamma delta T cells because you need a plastic surface for the monocytes to stick down and be able to activate, so they'll release the isopenyl pyrophosphates in response to zoledronate and then activate your cell product. We showed on the flow cytometry plot to the left, you see the T75, and to the right you see the bioreactor, and actually we doubled our production percentage of gamma delta T cells with this bioreactor. All of our cell products showed cytotoxicity against AML cell lines, even some of the more resistant ones, and all three experiments showed that we would have sufficient cells to begin a transplant procedure. So then you go to the pre-IND meeting, and the first thing you do is you read the guidance, because that's really frustrating to the FDA when you don't do that, and you don't want to frustrate the FDA because they want to be your friend. Data and supporting primary documents, flow cytometry, cytokine analysis, PCR, sequencing, all your signed date and date stamped material that you started when you were thinking about this as a postdoc. Animal model safety of efficacy data if necessary. This study didn't require it because a lot of this was already published, and we were able to just show the work in papers. But if your animal facility is not GLP compliant, that's not going to kill you, but you have to show the FDA the areas of non-compliance, how you've overcome those, and how this affects your project. A lot of academic animal facilities aren't GLP compliant. Comprehensive but not excessive supporting literature package. Just because you send a list of 180 references does not mean the FDA is going to look them up when they want to see what you're referring to. And a draft clinical protocol, a draft CMC, and a validation plan. So in the pre-IND meeting, the FDA is not going to tell you how to do the trial. You're the expert. They're responding to specific questions that you have that are related to the regulatory guidance. Do your homework, read the guidance, and align your questions to the guidance where you see there might be a bit of a discord. Gather your team. You want to have everybody there to help you write this, a PI, the cell therapy lab director, research nurse specialist, quality manager, consultants, and anyone else that's central to the project. Discuss and review the document before you send it in, and then the FDA will respond to you with answers to your questions. And when you get there, then you make a decision as to whether you want to do an in-person meeting, like having everybody fly up here to sit around the table, a teleconference, or just say, thank you guys very much, this is sufficient, we can move forward. I always take the middle ground and do the teleconference. I think it helps get everybody acquainted with each other and also resolve any outstanding questions. So our pre-IND discussion, I can tell you about what we talked about. We asked about the fact that we were using approved drugs and other reagents and containers in GMP grade. We asked if they suggested further validation of these materials. We asked if there were any additional toxicology or animal studies that were necessary in addition to the literature that we had submitted to them, safety or risk management concerns, and whether or not they would rather us do this in our institution or perhaps take it off-site, and what the ups and downs were for that. So that, and received satisfactory replies. Then you learn that you have to find a way to fund your dream, and it will not be easy. Clinical trials, standalone R01 and U01s are notoriously difficult to obtain, and all of the University of Alabama's cancer enterprise we have. And that's a neuro-oncology oncolytic virus, R01. Funding for a GMP scale-up and validation even more so. People don't want to give money, the taxpayers don't want to give money for you to play in the lab, and sometimes the companies don't want to either. So you have what is commonly known in our parlance as the Valley of Death. Now, my experience on this, I submitted a primary and an A1 for a clinical trial R01, and I got 31, 32. Nothing really you could see that you could really fix from the reviewer's comments, but you try, you submit it again, you get 31, 32, which they tell you really like your idea, but we're never going to give you the money. So then we went looking for a company to work with us, and we're going to talk a little bit about that as well. You may be a great scientist, but if you have a PhD, I was a little luckier because I was a nurse practitioner before I came into this field, so I had some clinical knowledge and ability. But if you're a PhD scientist, you're probably going to have to give up most of the control and probably most of the glory too. Like it or not, a physician is going to run your trial. They're probably going to give the big talk at ASCO when you start seeing your fabulous results. Believe it or not, there are a lot of people that peel off at this stage and go back to the lab and try to discover something else. But if you want to stick it through and share, this is an opportunity to get your work out there. And once you license your IP and receive industry funding for scale-up in phase 1, you've lost control. You're done. They're taking over. And if you think this might be a problem, quit and work for the company. Who knows? Who knows? You might become chief scientific officer. Lesson 8. The process of scale-up, validation, and standardization is long, difficult, tedious, very expensive, and occasionally infuriating. You have intimate familiarity with 21 CFR 1271, 312, 210, 211. Process development, media, cytokines, beads, all of that has to move from lab to clinical. The formulation will change. You're going to spend $20,000 a run and then find out that somebody did something wrong and then throw the furniture around your office, which I did several times. Process validation, make sure it works. All three, at least 2 or 3 times, you can repeat it. And again, the sterility, potency, purity, and identity. Our process flow, I'm going to go through this quickly, but basically what you show the FDA is the mapped out beginning to end in your bioreactor. These are canned Miltany culture and setup and alpha beta T cell depletion programs that they'll give you. So for this particular project. So getting a little close on time here, but this is our trial. We start with we condition the recipient to start with a haploidentical donor, do a non-mobilized apheresis, activation of gamma delta T cells and expansion, cryopreservation. Then the donor comes in for a mobilized apheresis, produces a graft which is then given to the patient. When the patient has neutrophil engraftment, the gamma delta T cell graft is frozen and infused. So once you have a process defined and your clinical protocol completed, you're ready to write your IND. You can still use a paper submission process if you're in an academic institution. The FDA offers multiple guidance documents. The one that's sitting up there is one of my favorites. Consultants can be helpful. Always have it reviewed by somebody who may not be all that friendly to you. So supporting documentation, I'll just suffice it to say that there's lots of it. And lots of appendices, particularly with results, with respect to your certificates of analysis, batch records, testing, those types of things. There's a 30-day review process. You can't start until that's over. You're very likely going to receive one or multiple phone calls from the FDA requiring clarification. If you're placed on clinical hold, you'll be informed prior to the 30-day review. And approval can be a simple email with one or more detailed follow-up in 1 to several weeks. This was the little one-liner that we got that approved our IND. And the best part of this is once you finish the bottle of champagne, you realize that you're only at base camp. So I want to thank the entire team that did this from 1992 to 2018. Lots of folks contributed. I notice I never use a personal pronoun with these because I can't claim much credit for it. These guys were just really great from several institutions. Several funders, companies who helped as well, and of course Incisys, who is now running this file, and his CEO, William Ho. That's all I got. If anybody's got any questions, and Rick will let anybody ask any, I'm here to answer them.
Thank you, Larry. We'll take one question. I just want to say, you know, you threw out a lot of lessons, but I think the most important one for the audience, resiliency.
Yes.
You have a good idea. You're going to suffer setbacks. Don't give up. So we do have time. We can take one question. Well, let me ask you, real quick. So at some point, right, you have a great idea. You're going to have to partner, and your choice in partners, how important do you think that is?
I think it's very important. I mean, a lot of it is just simply what's the synergistic ability between you and the partner, and also you want the partner to tell you where they plan to take this and how they're getting the patients, how it relates to the rest of your work. But also you need that person or that company to be able to see your work proceed. I think that you cannot have a big ego and do this. You have to be able to put the patient first and understand that you are part of a group that is ultimately going to make somebody's life a whole lot better.
Okay. All right. Well, thank